Recording. There we go. People online, do you all have any questions so far about anything that's going on? We've talked a little bit about how to study. We're going really fast pace. You know, chapter a day, we're already on page 300 something in our book. So questions about anything to this point? All right, cool. So as we go through today's lecture, please just say, can you repeat that, or can you explain that again if you'll have any more questions? I'm going to go ahead and share my screen. Presentation mode, let's get our pointer, laser pointer, and then go down. Okay, so I posted, let's get our projector on. So you'll have the chapter 9 slides on Canvas. The first slide here, once our projector turns on, is the rubric for your paper. So we are going to take our exam a week from tomorrow. So your exam's coming up real soon, then we're going to go on our little break for Christmas and New Year's, come back, and then at the end of week 4, the week that we return, this paper is going to be due. So you can have that break, but if you don't want to do it, you got the week that you come back to get it done, okay? What I've done is I've created a general rubric. You can follow this, but you don't have to follow it to a T. I want this to be, this is a paper for you all, okay? If you all are going to work in healthcare, I want you to understand the pandemic now, as opposed to learn about it after it's over. So use this as a way to explore whatever you're really interested in. All right, you're going to answer 5 main questions. I want you to tell me something about the SARS-CoV-2 virus, the virus that causes COVID-19. So some questions that you can answer are, what is a coronavirus? Where did it enter the human population? How is it transmitted? Just general things about the virus. I want to know about the disease. So what does the disease look like? Who gets it? What are risk factors? Who's more prone than others? And again, you don't have to answer all of these. Just answer some of them. Answer the main question that is numbered. The third, what are the government mandates? What does the NIH, the National Institute of Health, the CDC recommend that we do? What does Kentucky recommend we do? What are the differences between states? If you want to take it that direction, talk about that. Why are the mandates important? What happens if people don't follow them? What happens if people do follow them? Like, why do we wear masks? Why are we all in here with a mask on? Why do we sanitize our venues? Tell me about it. I want you all to go into the literature and find a paper, so either an experiment or a clinical study that looked at mask wearing, vaccine development, just anything in the literature. And so if you go to your library page, you should be able to find research guides, and we're going to look for something called PubMed. Have you all heard of this before? Or looked at your research guides? No? Okay, so PubMed is where you're going to go to find any sort of a paper like this. It's like a Google for science journal articles related to health. So if you just go into your research guides, you'll have free access to all this stuff. Google Scholar is also another option. And then I'm interested in your predictions about the pandemic. Do you think it's going to be over in a year? Do you think we'll be wearing masks 5 years from now? What do you think and why? I'm genuinely interested. So 1 through 4 is worth 20 points, 5 is worth 10 points. You're going to get 5 for your formatting here, so 1-inch margins, 12-inch font. To keep your text to a full 3 to 5 pages, we're going to do a cover page with your title and your name. So your text will start on page 1, right? You have cover page, page 1, 2, 3 to 5. And then you'll put your references on a separate page. So this, I'm thinking maybe a day. You could do this in a weekend. I'd like to see some effort. Again, this is for you to explore and learn now. This is a really valuable opportunity for you. If you're really struggling or if you just have questions, get a hold of me. Let me know what you need. I'd be happy to help you. All right, so let's talk about chapter 9. We are talking about how to deal with microbes today. And then we're going to talk more specifically about some antibiotics tomorrow. Okay, so there's a difference between sterilization and sanitation or sanitization. Sterilization is the complete removal of anything that's alive, including viruses that aren't even alive. We're going to get rid of them, though. So bacteria, spores, viruses, everything is dead under sterilization. Nothing is going to survive. But as soon as you sterilize something and then expose it to the air, now it's prone to contamination. So sterilization versus sanitization. Sanitization is just a reduction in the number of disease-causing organisms or pathogens. So reduction versus complete elimination. That's the difference I want you to know. So sanitization is the reduction in the number of pathogens, whereas sterilization is the complete elimination. Everything is dead. Everything's gone. So if you're about to do surgery, you don't want to sanitize your surgical equipment. You want to sterilize it. That is a big difference. You don't want there to be hardly any chance of your patient getting sick. So we would call it contamination.
Okay.
So contamination is just going to mean you're going to get, in our context, a microbe potentially on a surface or a substance in a liquid that you don't want it on. So if you have something present that you don't want, we would call that contamination.
Okay, thank you.
You're welcome. All right, and then within a hospital setting, a healthcare setting, if you get an infection in that setting, we call that a nosocomial infection. So MRSA, methicillin-resistant Staph aureus, is a common nosocomial infection. It lingers around hospitals, and patients can get it. If they get it in that healthcare setting, it's a nosocomial infection. So again, we want sterile equipment, not sanitized equipment, if we're going to open somebody up. But we can't sterilize everything. You can't sterilize a bed very easily, right? But we can reduce the number of pathogens on it. All right, so just to lay out some terms here, we can have agents that are microbicidal. That just means that we are going to kill a small living organism. Microbes, small things that are alive. Could be a fungus, could be a bacterium. If it is an agent designed to kill bacteria, we'll call it bactericidal. If it's an agent used to kill a fungicide or a fungus, we'll call it fungicidal. So both of these fall under the category of microbicidal agents. So that is if we kill organisms, but we could also just prevent their ability to grow. So we might not kill them, but we can reduce their numbers and prevent their ability to grow into more organisms. These are going to be microbiostatic agents. So something that interferes with DNA replication or protein synthesis. These won't directly kill an organism, but it won't allow it to function properly, or it won't allow it to divide properly, so it'll end up dead.
So is that what we use in the sanitizer?
If we are sanitizing, I'm going to say we're probably going to want to go for microbicidal agents. That's going to be more effective than preventing it from growing. We want to kill something. Yeah. And again, we can have bacteriostatic or fungistatic, just referring to how specific it is. Yeah. So I would say, we could have both. It depends on what's in the sanitizer. So ethanol will disrupt cell membranes and kill bacteria. But if you don't get all the bacteria, you might not kill all of them. So I'm going to say, depending on the agent, one agent could be microbicidal and microbiostatic. I would need to look into that. But with hand sanitizer and ethanol, I would say that is more on the microbicidal. We're going to kill organisms with that. Yeah. But I follow you where you're saying if you kill 99.9%, what about that little bit that survives? If the alcohol prevents their DNA synthesis, or if it throws off their metabolism by destroying proteins, then we would call it microbiostatic. So I think ethanol is a good example of one that would probably fit into both categories. Yeah, and I'll look into that. We'll talk about that tomorrow. Really good question. Okay, this is a figure that lays out what we're going to talk about for the next 15 minutes or so. So all of these ways, heat, filtration, radiant energy, or low temperature, these are all ways that we can physically control microorganisms in the environment. We're going to start with heat. You'll notice here there's a difference between dry heat and moist heat. So if we use heat, dry heat specifically in this case, so a flame from a Bunsen burner is going to denature proteins, destroy your nucleic acid, so your genetic material. It also removes water. Water is essential for life. If you destroy the proteins, the DNA, get rid of the water, these microbes are dead, okay? So you're always going to do this before you go into a culture with your bacterial loop. You will always heat your loop over a flame. It's going to glow orange. Then you're going to pull it back, let it cool, because remember, microbes are sensitive to temperature. You don't want to put a glowing orange loop to get bacteria. You're just going to kill them. So you got to pull it back, let it cool, but you're worried about contamination. So you keep it close to the flame. It's just the game that you're going to figure out, I promise. How much heat, how much time after heat. This is really practical for your lab setting. It'll make more sense. So we call this incineration, use of direct flame to kill microbes. This is an image of livestock that have been burned. The book mentions a pathogen that was just spreading like crazy through livestock, and the only way that they could figure out to get rid of it was to just burn all these animals. Really sad. I think they said something like millions of animals.
Nick,
Probably some happy soils.
Considered dry heat.
So dry heat is something like a flame or an oven.
Oh, gotcha. Oh, that's
Yep.
really sad.
It is really sad. So if we're talking about heat, we have some definitions that we can apply. The thermal death time. This is going to be the time that it takes at a certain temperature to kill organisms. And then related to that, we have the thermal death point, which is the lowest temperature required to kill organisms in 10 minutes. So thermal death time, we have a specific temperature. What is the lowest amount of time to kill microbes? Thermal death point refers to the temperature used to kill microbes in 10 minutes. So take a look at this figure here. So on the x-axis, we have time in minutes. On the y-axis, we have number of surviving cells. How would you describe surviving actively dividing cells? What is the one word that we're going to use to describe dividing cells? Starts with a V. Think greenery. Vegetative, right. So these are vegetative cells. Otherwise, we would call them dead cells or spores. So we have cells surviving as a function of time. We have three different species, let's just call them here, bacterium A, B, and C. If we expose them to heat, which of these would you say is the most susceptible to death by heat? A, B, or C, and why? I would say A. I agree with you. Why? To do what? Yeah, so species A, so in all cases, we start with 10 to the 6 bacteria. Species A, we eliminate all these bacteria in 20 or so minutes. After an hour, B is dead, but we still have about 1,000 cells per whatever volume of strain C. So we could say that A is the most susceptible and C is the most resistant to death by heat. Just being able to look at a figure and interpret it. You all should expect to see something not exactly like this, but similar on your exams, where I'll give you a figure that you may or may not have seen, and then I'll ask you a question like this. Which is the most susceptible? Which is the most resistant? All right, so with dry heat, we can have things like an oven or a flame from a Bunsen burner or just, any sort of direct fire. Moist heat is different. This is going to be a little bit more effective in a lab setting for our purposes. So moist heat, like boiling water. So we can do this faster than dry heat most often as far as killing microbes. We're going to denature proteins just like we did with the flame. But keep in mind that boiling water won't kill spores, and it's not always going to inactivate viruses. I think when we talked about spores, the book said you have to boil water for, I think it was 2 hours to eliminate spores. Which, during our water advisory warnings, we're boiling water for 10, 15 minutes, not 2 hours. So what you're going to see in the lab, this is going to be in any lab, any hospital. This is how we're going to clean our surgical equipment. We're going to use an autoclave. This is a figure showing an autoclave. We're going to use high pressure, high temperature, and steam. So water vapor. And that high temperature water vapor is going to be used to kill anything that's alive. So all of our growth media that we're going to make, our broth, our agar for our petri dishes, all of this is going to be sterilized through an autoclave. The book mentions a pre-vacuum autoclave. I've never used one of these, but what this works is it's going to decrease the pressure by sucking out all of the gas that's present in this chamber before the temperature rises. So it increases pressure before temperature also raises. Okay, so think about this. Pressure and volume are inversely related. So if you suck out the gas from an autoclave, you increase the pressure. And you would use this if you had something that was sensitive to heat. This is going to happen faster. We can autoclave in about 4 minutes with the pre-vacuum autoclave. We need about a 15-minute cycle for the regular steam autoclave. And that time is at temperature. So it'll take time to go up. You're going to hold it for a given amount of time, and then we have to decrease the pressure over an amount of time as well. So the whole cycle might take an hour, but you'll be at that high temp, high pressure for 15 minutes in a normal autoclave. There's a process called intermittent sterilization or tyndallization. This is going to require 3 days. Expose them to high heat for 30 minutes. Then you're going to put whatever it is in a growth chamber. And let's say that you killed everything except the spores. Overnight, those spores will turn back into vegetative cells, and they'll be present for you in the morning. So you hit them with another 30 minutes of heat, put them back in a growth chamber. You should have killed all the spores, but let's say some survived, and then those turn back into vegetative cells. On day 3, you come back and do the same thing again, 30 minutes heat, and that is the intermittent sterilization. So you do it 3 times. But I'll just go ahead and say the normal autoclave will kill endospores. So this is very safe.
So what would you use?
I've never used it. Yeah, I have always just used a normal autoclave. And everyone that I know has done that, or filtering. We'll talk about filters here in a second. So why would you do this? If you had something that maybe couldn't be exposed to high pressure, or if something couldn't be exposed to steam, I think is what the book mentions. Because the autoclave is going to use steam, this tyndallization technique, I don't believe does. I believe this is just an elevated temperature. I think they say things that contain oils, this might be useful for because oil and water don't get along. Not the most important thing on the slide though. Autoclave is the most important thing here. All right, so we are not gonna boil our food. We're not gonna cook our juices, but we will heat it to about 63 degrees for a half hour. Keep in mind that our bodies are 37 degrees. Water boils at 100 degrees, so we're going to go somewhere in between there. What do we call pathogens that are going to infect us so they can survive within the temperature range that humans are in? Right, mesophiles. And so mesophiles are going to survive up to about, I think it was 45, 46 degrees Celsius. So if we can heat our foods, our juices, our milks up to 63, we're going to get rid of most of the pathogens that would hurt us. Not everything, but most of it. I have some friends from Nepal, and apparently Nepalese people drink raw milk, right? I mean, people in the US drink raw milk too, right? But tuberculosis can be transmitted through milk. So apparently a lot of Nepalese people end up dying from tuberculosis infections later in life. So now, like, our generation is pasteurizing milk. So be aware that cross-culturally, different populations of humans are going to control microbes at different time periods. And there's no like right or wrong way. We're just trying to be safe. We can use a filter. So you all have used filters before. You know how this works. You're going to trap things by not allowing them to pass through small pores. Okay? We call this a membrane filter. So we have